In this video, I'll focus on the first three objectives on this slide, starting with hematopoiesis, and I'll end with presenting hemoglobin. Let's start with definitions. It's always important to understand terminology before a new topic is introduced. Hematopoietic or hemopoietic refer to the same thing, and that is the forming of new blood cells in blood. Your body continually makes new blood cells to replace old ones. The yolk sac is a special structure associated with the developing fetus. Inside the yolk sac, a process called hematopoiesis happens, which is when one's very first blood cells are made. You can see where the yolk sac is in regards to the embryo in both the illustration and ultrasound images below. A hemocytoblast is a stem cell from which all other types of blood cells are made. A hemocytoblast is said to be pluripotent because that one cell type is capable of producing many other different types of cells. So in order to make blood cells, tissues that are capable of the tasks are needed. These tissues are hematopoietic. As already mentioned, this starts very early on during embryologic development. What is meant here that stem cells colonize tissues is that the production of the stem cell begins in the yolk sac, and then these cells are delivered to different parts of the embryo, specifically the bone marrow, liver, spleen, and thymus. The stem cells play an important role in healthy development. The thymus plays a crucial role in helping our bodies fight off illnesses. Inside the thymus, special cells called T cells are made. These T cells are like the soldiers of our immune system, helping to identify and destroy harmful bacteria and viruses and keep us healthy. During development, the thymus helps these T cells learn to recognize the difference between our own body cells and harmful pathogens. The red bone marrow is the site of hematopoiesis throughout our lifetime. I'll discuss the different cell lines individually, as well as erythropoietin, thrombopoietin, and other colony-stimulating factors, and their role in the production of the various blood cell types. This is an illustration of the hemocytoblast, labeled here as the hematopoietic stem cell, right at the top. You can see the different lineages or cell lines that result from the stem cell. The lineages are as follows. The myeloid line forms the red blood cells, monocytes, all granulocytes, and platelets are derived from, and the lymphoid lineage from which T cells and B cells originate. This is a simplified diagram and a good place to start when learning how these cells develop. The blood vessel is represented in this image so you can see which cells travel via the blood, and you can also see which cells will leave blood circulation to reside in other tissues. Mast cells are found in connective tissue, especially under the skin. T cells and B cells reside in bone marrow, and monocytes circulate in the blood for a few days before migrating to body tissues. An example are the microglia you learned about in Bio 201. This illustration shows more details and includes the lineage that leads to the formation of platelets, which was missing from the myeloid progenitor line in the previous slide. I have outlined it here in red. Now let's get into the details of how red blood cells are formed. This image gives greater detail about the erythroblasts. The line follows like this. The first recognizable cell in this line is the proerythroblast. Then come stages for the erythroblast to develop, ending with the normoblast. Next stage is the formation of the reticulocyte. At this stage, the cell enters the bloodstream. Erythropoiesis is the term that specifically refers to the production of million red blood cells, RBCs. There are 2.5 million RBCs produced per second. The production site is in bone marrow, and their development takes up to 5 days. Erythropoietin, which is produced by the kidneys, is the messenger that stimulates RBC production. The proerythroblast turns into the erythroblast. You may recall that blast refers to an immature cell, whereas site indicates a mature cell. Erythroblasts are capable of hemoglobin synthesis. They multiply and transform into normoblasts. At this stage, the normoblasts discard their organelles, including the nucleus, leading to the formation of reticulocytes. Reticulocytes leave the bone marrow and enter the bloodstream, where it takes 24 hours for their maturation to be complete, after which they are termed erythrocytes. The very special function of erythrocytes to deliver oxygen efficiently is accomplished by the globin protein hemoglobin. Hemoglobin is made of 4 protein chains, 2 alpha and 2 beta chains. The important thing is that each chain contains an iron-containing heme group. Oxygen reversibly binds to the heme group. Since there are 4 heme groups, each hemoglobin can bind 4 oxygen molecules to transport. Each red blood cell holds about 260 million hemoglobin molecules. As already mentioned, iron is essential for hemoglobin production. Once iron gets absorbed from the digestive tract into the blood, it binds to transferrin to be transported. Iron is stored in bone marrow as ferritin and as ferritin in the liver. In addition, mitochondria require iron for proper metabolism. Other nutrients required for erythropoiesis are B12 and folate, which aid in cell division. Vitamin C assists with iron absorption from the intestine, and copper is needed for iron to be transported from the cells to the blood. This diagram illustrates that iron is needed in the diet. It is absorbed by the small intestines, moved through the body by transferrin, a transport protein. Most absorbed iron is used by red blood cells to make hemoglobin. Some is stored, and the rest is used for other processes. For example, iron is a cofactor for proteins involved in DNA replication and repair. The stimulus for erythrocyte production occurs when low levels of oxygen are detected by receptors in the kidneys, liver, and carotid bodies. Especially important are the kidneys when they detect low oxygen levels. They increase production of erythropoietin. This stimulates RBC production in bone marrow, but this is not exactly a quick fix, as it takes 3 to 4 days for the red blood cell count to increase. Remember, hormonal action sometimes takes time.